So my presentation is basically we looked at Mendelian randomization, which is a computational statistical method. And basically what this does is it looks at basically what alleles within the genome at sites called QTLs. So this is quantitative trait loci. And these are basically these are SNPs within the genome, which basically they alter molecular traits, the amount. So for instance, an eQTL, which is an expression QTL, this basically a SNP, one of these QTLs would either cause expression or to increase or decrease, depending from SNP to SNP, it varies. So we looked at expression QTLs, protein QTLs, which is protein abundance, the levels of proteins fluctuates, and splicing QTLs. And again, it's just the number of splice variants would vary it. So what we did is we took in MR, basically you need two data sets. We use two sample Mendelian randomization, and you take what you call the exposure and the outcome. So the outcome is that's kind of your phenotypic data. This is, you would either have people who with glioma, with brain tumors, or you would have controls. And we have around 30,000 patients in total. 18,000 of them are controls. Around 12,000 of them are glioma patients. Around 6,000 are GBM, which are grade 4 tumors, which are kind of the worst of the worst brain tumors that you would expect to see. And the others are well, we turned them into one group of non-GBM, but it was astrocytomas and oligodendrogliomas. Still really horrible tumors, but not as severe as GBMs. So you have phenotypic data on those patients, and then you have an exposure data set, which is your QTL data. And basically we use GWAS, so that is genome-wide association studies. And basically we use three different data sets, one for each QTL, and these QTLs are they're derived from big data sets as well. And all of ours have come from tissue. So one of my colleagues, Lily, she uses MR as well, but she uses blood. So she uses a different type of she gets different biomarkers because it comes from different things. So basically, what you do is MR looks at the alleles in patients across the two groups, and you can try to determine whether the allele at a specific QTL causes the risk of glioma to increase or decrease, depending on whether they have the reference allele, so what kind of the majority of the population has, or the effect allele. And this effect allele is the one that causes the level of expression or splicing or protein abundance to vary. So we did this. We did the eQTL, the SQTL, the pQTL. We ran the MR. We did some sensitivity analysis, just basically to make sure that I mean, MR is a really good method because it detects, it kind of reduces bias anyway. It's really good for that. The only thing with MR is there is 3 associations, which you kind of have to follow in MR, and that's your genetic instrument, your IV instrument of variable is associated with the exposure. So is the IV exposed to the QTL? Is it associated with the QTL? There should be no confounding factors between the IV and kind of the exposure and the outcome. Try and minimize. It's obviously impossible to completely remove all confounders, but you try and minimize it as best as possible. And that the third assumption is that the IV is linked to the outcome only through the exposure, and there's nothing else that's kind of it can be linked with. So it's a really good method, but I guess it does have its pitfalls that if there is confounding, it can kind of ruin your analysis a little bit. Anyway, so Yeah, so we did our MR, we did our systematic, our sensitivity analysis. So this was what did we do? We did conditional joint analysis, we did genetic co-localization, and we did Steiger filtering. And these are basically just to make sure that the results that we get are, is it the right direction? Is the so Steiger filtering tests for directionality. So basically, is the QTL causing the change in phenotype or the cause and variation of the QTL? Because if it's the wrong way around, you don't want basically it to be the other way around. It's all about one directionality. And then the co-localization. So a lot of SNPs are in high LD with each other, so they're in linkage disequilibrium. So the way MI works is it uses proxy SNPs. So you use a SNP as a proxy, as a marker, and essentially what you do is you want to make sure that you can because so many instruments are in high LD with each other, you can kind of reduce the amount of instruments by using LD. So that's what genetic co-localization does. I didn't talk about that in my presentation just because it was such a quick presentation. So my results, so the eQTLs, we had a really nice data set with some pretty, for glioma, because glioma data sets are quite small, you'll find anywhere. We had a pretty nice data set, especially with, we had 5 tissues, one of them was cortex data, and that was around, I think it was around 2,000 patients, which is pretty good actually for glioma. Some of the smaller data samples were kind of like the hippocampus was only a couple of hundred. And what we found was we found 20 different genes which had some sort of variation with glioma, basically the risk of developing the risk of glioma. And then we split these by either all of the glioma patients together, so they were all combined. They were just GBM, so just the glioblastoma multiform, or the non-GBM, which is the group that I said earlier about the 2 combined groups. And it was quite interesting because I think what we found is for some, you could find a gene where, for instance, in one tissue, let's say the basal ganglia, you might have a result where in one group, it reduces the risk slightly, but in another group, it slightly increases the risk. And then we found that this could also vary from tissue to tissue. So one gene in one tissue might decrease the risk, but in another tissue, it might actually increase the risk. So that was really interesting. And then what we did was we took our results and we looked at a meta-analysis, which was by I think it was Ben Kinnersley, and this is a big systematic review meta-analysis on kind of what we know are risk factors, genetic risk factors for glioma, and we said, okay, let's kind of rule out any genes that already come up in the literature. So these are your common ones, like kind of like TERT, ARTEL1, kind of these, things to do with telomere length and telomerase, and then things like CDKN2A and 2B, very common markers in GBM. And we also kind of removed anything which was at the same gene locus, because like I said, MR relies on LD. So if you've got a marker which is in really high LD, or it's in a shared location with a gene which has already been discovered, it might be that actually that marker is in LD with that gene. So we removed those as well, just to kind of try and they still cause an effect, but we just wanted to look for novel targets. So we removed those, and we found was it 3? I think it was 3 new targets within the eQTLs. And like I say, there's a lot of variation in the odds ratio. So I think in the eQTLs, the risk ratio went down to about 0.5, reduced risk. And then up, this is mainly, I think, TERT and RTEL kind of increased about 7-fold, which is what you'd expect. So we did the same thing in the SQTL data set. The SQTL data set, the samples were a lot smaller. So there was more tissue. So it was better that we had more granulated tissue samples. So whereas in the eQTLs, we had 5, it was like cortex, cerebellum, very broad regions of the brain. Whereas we had 14 different tissue samples in the SQTLs, and these are more kind of niche. So there was like the amygdala and specific areas of the cortex. So we looked at like broad areas. And we did find some associations, but there wasn't as many, nowhere near as many. There's only 3 genes we picked up from this. And these all seem to be protective of glioma as well. They all kind of reduce the risk of glioma. And there wasn't as many. And what we found as well is that none of the associations were purely in GBM patients. They either seem to be in the non-GB portion of patients or in the merged data set. And the variation of the odds ratio was a lot smaller. They were a lot more packed tight compared to the eQTLs. The pQTLs, we didn't find anything. None of our findings, we used Bonferroni corrected p-values purely because it's just, if you use a normal kind of that threshold of 0.05, you would have so many different instruments. So we try and reduce this by using a Bonferroni corrected p-value. So yeah, that was a bit disappointing because I guess with pQTLs, this is kind of where the really good drug targets come in, especially with high risk patients. So yeah, we didn't find anything in the pQTLs, but we'll come back to that. So anyway, across this analysis, across the SQTLs and the eQTLs, we found 4 genes which had never been implicated, nor had their locus, the region that they were found on. So we kind of did some enrichment analysis and looked at what these genes did, what they were involved in, how they work. And off the top of my head, I can't remember them all, but they tend to be this common thing of, it was all to do with immunity. And kind of, I remember one of them was to do with T cell activation, I think, or maybe it was B cell, I can't remember. No, it's been a while since I've looked at the enrichment analysis. But one of them was a gene called GAL-NT6, and this gene is it's associated with I think in lung cancer it's been found to be associated with invasion and metastases, and it's got an association with GRP78, which is a protein that we see quite commonly upregulated in glioma cells. So this is quite an interesting finding. I spoke to one of my colleagues who she does a lot of wet lab things, and she's been working with cells with GRP78 upregulated. So that's something. So I guess our next steps now after that, where we're going to try and find a new SQTL data set with bigger samples, which we know there is one coming out soon. When, we don't know, but it is coming. So do a validation data set with that SQTL. And then we have been given access to another pQTL data set, which hopefully we find something in this one, because this is kind of the big data set that we want to find something in, especially for drug discovery. pQTLs are the kind of the big thing. So once we get full access to that, we're going to start doing a similar kind of thing with that, and hopefully find some pQTL results. And just I think with any epidemiologist will tell you, you can always do more over and over again with bigger data sets and just validate your findings. And I guess it's just the same. Yeah, we just need to keep going and see what we can find, basically.